Our lecture is polymerase chain reaction (PCR). PCR was invented by Mullis in 1983 and patented in 1985. Its principle is based on the use of DNA polymerase, which is an in vitro replication of specific DNA sequences. This method can generate tens of billions of copies of a particular DNA fragment from the DNA extract or template. If the sequence of interest is present in the DNA extract, it is possible to selectively replicate it in a very large number. The power of PCR is based on the fact that the amount of matrix DNA is not a limiting factor. We can therefore amplify nucleotide sequences from very small amount of DNA extract. So PCR is therefore a technique of purification or cloning. Application of PCR. It is a technique now essential in cellular and molecular biology. It permits, especially in a few hours, a cellular cloning of a DNA fragment through an automated system, which usually takes several days with a standard technique of molecular cloning. PCR is widely used for diagnostic purpose to detect the presence of a specific DNA sequence of this or that organism in a biological fluid. It is also used to make genetic fingerprint, whether it is the genetic identification of person in a context of judicial inquiry, the identification of animal varieties, plant or microbial for food quality testing, diagnostic or varietal selection. PCR is still essential for performing sequencing or site-directed mutagenesis. Finally, there are variants of PCR such as real-time PCR, competitive PCR, PCR in situ, etc. Application of PCR. The revolutionary evolution of the molecular biological research are based on the PCR technique, which provide the suitable and specific products, especially in the field of the characterization and the conservation of the genetic diversity. Several applications are possible in downstream of the PCR technique. First, the establishment of a complete sequence of the genome of the most important livestock breed. Second, development of the technology measuring scattered polymorphism at loci throughout the genome, example SNP detection method. Third, the development of the microarray technology to measure gene transcription on a large scale. The study of biological complexity is a new frontier that required high throughput molecular technology, high speed and computer memory, a new approach to data analysis, and the integration of interdisciplinary skills. Principle of the PCR. PCR makes it possible to obtain by in vitro replication multiple copies of DNA fragment from an extract. Matrix DNA can be genomic DNA as well as complementary DNA obtained by reverse transcriptase PCR from a messenger RNA extract, poly-A-RNA, or even mitochondrial DNA. It is a technique for obtaining large amount of specific DNA sequence from a DNA sample. This amplification is based on the replication of double-stranded DNA template. It is broken down into three phases: denaturation phase, annealing or hybridization phase with the primers, and an elongation or extension phase. The product of each synthesis step serves as a template for the following steps, thus exponential amplification is achieved. Principle of the PCR. The polymerase chain reaction is carried out in the reaction mixture, which comprises the DNA extract, template DNA, tag polymerase enzyme, the primers, and the 4-deoxyribonucleoside triphosphate DNTPS in excess in an buffer solution. The tube containing the mixture reaction are subjected to repetitive temperature cycle several tens of times in the heating block of a thermal cycler apparatus, which has an enclosure where the sample tube are deposited and in which the temperature can vary very quickly and precisely from 0 to 100 degrees centigrade by Peltier effect. The apparatus allow the programming of the duration and the succession of the cycles of temperature steps. Each cycle includes 3 periods of a few tenths of second. PCR reaction component, as we said, template, 2 primers to flank the target sequence, 4 deoxynucleoside triphosphates to provide energy and nucleoside for the synthesis of DNA, buffer system containing magnesium, and DNA polymerase enzyme. This is the PCR component: DNA sample, primers, nucleotides, tag polymerase, and mixed buffer. This all put in PCR tube, which is put it in the thermal cycler for PCR cycle. The PCR cycle includes the denaturation and then annealing by primer, and third is the extension or elongation stage. Template DNA which contains sequence to be amplified, can be chromosomal, plasmid, linear, or circular. Only a small amount needed for reaction. Primers to achieve a selective amplification of a nucleotide sequence from a DNA extract by PCR. It is essential to have at least one pair of oligonucleotides. These oligonucleotides, which will serve as a primer for replication, are synthesized chemically and must be based possible complementary with both ends of the sequence of interest that one wishes to amplify. One of the primers is designed to recognize complementary a sequence located upstream of the fragment from 5 to 3 strand DNA of interest. The other to recognize always by complementary a sequence located upstream complementary strand 3 to 5 of the same fragment DNA. Primers are single-strand DNAs whose hybridization on sequence flanking the sequence of the interest will allow its replication so selective. The size of the primer is usually between 10 to 30 nucleotides in order to guarantee sufficiently specific hybridization on the sequence of interest of matrix DNA. So the primer is a small oligonucleotide that provides the start point for DNA replication. One primer required for each strand in order to amplify double-strand DNA. Primers must be oriented facing each other in the 5 to 3 direction. Primers must be complementary to template sequence targeted for amplification. Buffer mimic physiological condition for polymerase to function. There is variable of some buffer component which can be alter success of the reaction, magnesium concentration required for function of polymerase, pH of final concentration, NaCl concentration. Optimize condition for PCR using buffer that vary in these components. DNA polymerase, purified bacterial enzyme that catalyze DNA replication. Tag polymerase, isolated from thermophilic bacteria, can withstand high temperatures and repeated heating and cooling. Tag polymerase is a thermostable DNA polymerase named after the thermophilic eubacterial microorganism Thermus aquaticus, from which it was originally isolated by Chen et al. In 1976. Its name is often abbreviated to TAG or TAG-all. It is frequently used in PCR. Tag polymerase was identified as an enzyme able to withstand the protein denaturing condition, mean high temperature, required during PCR. Therefore, it replaced the DNA polymerase which previously used from the E. coli originally used in PCR. This is the tag polymerase and this is the DNA. The reaction of PCR is down into 3 steps per cycle, as we said before, which is denaturation, annealing, or hybridization. Third stage is elongation or extension. The denaturation, it is the separation of 2 strands of DNA obtained by raising the temperature. The first period is carried out at a temperature of 95 degrees centigrade called the denaturation temperature. At this temperature, the matrix DNA, which serve as matrix during the replication, is denatured. The hydrogen bond cannot be maintained at the temperature higher than 80 degrees centigrade, and the double strand DNA is denatured into single strand DNA. Second phase is annealing or hybridization. It is carried out at the temperature generally between 40 and 70 degrees centigrade called primer annealing temperature. Decreasing the temperature allows the hydrogen bond to reform and thus the complementary strand to hybridize. The primer shows single-strand sequence complementary to the region that flank the DNA to be amplified, hybridize more easily than long-strand matrix DNA. The higher the hybridization temperature, the more selective the hybridization, the more specific it is. Third phase is elongation or extension. This period is carried out at the temperature of 72 degrees centigrade called elongation temperature. It is the synthesis of the complementary strand at 72 degrees centigrade. Tag polymerase bind to primed single-strand DNA and catalyze replication using the deoxyribonucleoside triphosphate present in the reaction mixture. The region of the template DNA downstream of the primer are thus selectively synthesized. In the next cycle, the fragments synthesized in the previous cycle are in turn matrix, and after a few cycles, the predominant species corresponds to the DNA sequence between the region where the primer hybridized. It takes 20 to 40 cycles to synthesize an analyzed amount of DNA, about 0.1 microgram. Each cycle theoretically doubles the amount of DNA present in the previous cycle. It is recommended to add a final cycle of elongation at 72 degrees centigrade, especially when the sequence of interest is large, greater than 1 kilobase, at a rate of 2 minutes per kilobase. PCR makes it possible to amplify sequences whose size is less than 6 kilobase. The PCR reaction is extremely rapid. It lasts only a few hours, from 2 to 3 hours for a PCR of 30 cycles. Thermocycle. PCR machine or DNA amplifier is a laboratory apparatus needed to run the reaction of PCR. It's most commonly used to amplify segment of DNA via the polymerase chain reaction PCR. The device has a thermal block with holes where tube holding the reaction mixture can be inserted. This is the hole and this is the tube. Put it here. This slide shows the process of PCR technique. This is the target sequence of DNA. This is the first step, denaturation. In 95 degrees centigrade, the heat briefly separates the DNA strand here into two strands. Second step is annealing, cooling to the 55 degrees centigrade to allow primer to form hydrogen bond with end of target sequence in 3. This primer and this reverse primer here. The third phase is the extension. The DNA polymerase, tag polymerase, add a nucleotide here to the third end of each primer here. This the tag polymerase, and this the addition of nucleotide here to the third end of the each primer. This and this also here. This the elongation or extension phase. This slide shows the same process. This is the target sequence of the genomic DNA. In first cycle, the denaturation, heat briefly separate the DNA into two strands. Second, annealing, cooling to 55 degrees centigrade, allow primer to, this is primer and this, to form the hydrogen bond with the end of target sequence here and here. Third phase is the extension or elongation phase. The DNA polymerase add a nucleotide to the third end of each primer here and here. Second cycle yield 4 molecules. This 4 molecules formation, and cycle 3 yield 8 molecules. 2 molecules here, this and this one, match the target sequence here. So this is the end of PCR technique. Thank you for listening.